audio recording of the lecture on virus and host cell viral diseases. Slide number 2, eukaryotic host cell. In general, viruses can infect a host cell by initially binding to a cell surface receptor of the host cell. Cell receptors are located on the plasma membrane of the host cell and are protein or glycoprotein or glycolipid in nature. The host cellular metabolic machinery, the metabolic machinery of the host cell, provides the virus its replicative processes, meaning those processes that are necessary for the virus to double its nucleic acid or replicate its nucleic acid, transcribe its genes, and translate those genes into proteins. All of these are being provided by host cell metabolic machinery. Virus cannot do it by itself. Slide number 3, stages of viral replication. There are 5 major steps for the successful replication of a virus. The very first step is the attachment of virus to the host cell, which is done through host cell receptors on the plasma membrane. After successful attachment, virus gains entry or penetrates into the host cell. Once it penetrates into the host cell, it will go through the second phase, which is early gene expression. Early gene expression is usually done by early messenger RNAs, and they will usually code for non structural proteins. After early gene expression, we have the third stage of viral replication called genome replication, where the nucleic acid of the virus, either DNA or RNA, will be doubled and makes more of itself. Process of replication. After replication, we have late gene expression due to the transcription of late messenger RNA, and the translation of this will result into the production of structural proteins. Therefore, we have non-structural proteins of the virus, genome is replicated, we have structural proteins, and the last stage is the assembly of these viral particles to make new viruses, and these new viruses are being released from the cell. Usually, per one single virus that gains entry or penetrates into the host cell, 10 to the power 2 to 10 to the power 4 viruses will be released from the host cell. Slide number 4 is the pathogenesis of virus infection. Pathogenesis is the process by which the virus causes the disease. As we just discussed, the very first step during the infection is the entry of virus into the host cell. So virus gains entry. After successful entrance, virus undergoes through an initial incubation period, where I usually like to call it as setting up shop. So virus needs to find the exact tissue or organ that it causes the infection from. For example, flu virus needs to get into the nasal area for its infection. Uh, After that, virus will spread to other regions of the host cell and start replicating, making more of itself. And as a result of replication, sometimes local immune response will kick in the form of interferon to curb and control viral replication. If the local immune response is not successful, the disease will disseminate. So we have spread of the virus and the disease inside of the host cell. And at this stage, uh, host immunity will kick in. There would be a robust and effective immune response against the virus. It can be both in the form of cell mediated immune response and humoral immune response, which is the secretion of antibodies from terminally differentiated B cells called plasma cells. After the whole, so depending on the balance between the immune system and the viral infection, we either the host will resolve the viral infection, and there would be clearance of the infection, and patients will undergo recovery. But sometimes virus can overcome the immune system, and that will result in either a persistent infection or subsequent episodes of the infection. Sometimes death will ensue, and virus can be transmitted from an infected individual to a normal, healthy individual. Next slide. This slide is called virus infections, or some people call it patterns of virus infection. There are four different ways in which a viral infection can manifest its clinical signs and symptoms. The first would be asymptomatic, meaning that we have the disease, but we do not see the symptoms of the disease. A lot of individuals can be carriers at this stage because they are kind of a little negligence about their disease because they do not see any signs and symptoms of the illness. The next pattern is acute, where the patient exhibits the signs and symptoms of the disease. Next, we have persistent, where there is a long-term infection of the virus. And persistent can be divided into chronic or latent. Chronic infection is where we have an infectious virus, but we do not continuously see the clinical signs and symptoms of the disease. It's chronically there. Whereas latent is where we do not have any active virus replication going on. Virus resides in the cells and it becomes reactivated. Example would be Epstein-Barr virus, infectious mononucleosis, where virus resides in some cells. If the patient is under a lot of stress or immune stimulation, or other factors, will result in the reactivation of the virus, and a new disease will manifest itself. And lastly, the last pattern of viral infection is transformation, where the virus comes in integrates into the host cell genome and modifies host cellular processes. It basically modifies or deregulates normal host cell processes, such as changes in signal transduction pathways or changes in the dynamics of cell cycle regulation. This will result in the production of tumors. During transformation, there would be no infectious virus. We do not have an active viral infection going on, as opposed to the fact that virus has integrated into host cell genome, and those gene products will change or deregulate normal host cell processes. During transformation, we see viral DNA, whether it's complete viral DNA or partial virus genome. Next slide talks about sites of virus entry. Where are those regions of the body that are susceptible to viruses gaining entry? Viruses use or hijack these uh, mechanisms to get into the host cell. The very first site of virus entry is the skin or mucous membrane. These include mouth, nose, conjunctiva in the eye, oral cavity or nasal cavity, mouth and nose, uh, through scratches of the skin, creating an injury, through anus or urogenital skins and mucous membranes. Next, site of virus entry is through ingestion of the virally contaminated object through the oral cavity or mouth. Inhalation is the third site of virus entry through oral or pharyngeal area, so-called oropharynx region, or through the respiratory tract where we inhale the virus into the respiratory system, and maybe after that to the alimentary tract or digestive tract. 4 is direct injection through needle stick or through punctures in the skin. And lastly, viruses can gain entry through arthropod vectors. These arthropods serve as the vector for the virus, and when they sting a normal, healthy individual, the virus can gain entry. Next slide is a review of eukaryotic cell biology. This is a depiction of a eukaryotic cell. As we can see, we have plasma membrane on the outside, then a number of subcellular organizations. Some of them are in the cytoplasm. We call it cytoplasmic organelles, such as transport vesicles, Golgi apparatus, mitochondria, smooth and rough endoplasmic reticulum, cytoskeleton, lysosomes, and peroxisome, which are the garbage disposal of the cell. Or we have the nucleus of the cell, where at the center it has a nucleolus, and the nucleus contains chromatin or the genetic material, and nucleus is confined by a nuclear membrane that has some pores where some specific proteins such as transcription factors can shuttle back and forth between the cytoplasm and the nucleus. And I understand that since everybody has completed the prerequisites, so this would be kind of a review for everybody. The next concept is cellular gene expression. We have linear chromosomes. In humans, we have 23 pairs of it. And each, this is called diploid, each gene in the body is regulated by its own control elements. These control elements include a promoter and enhancers. Nucleus is the site of replication and transcription of the messenger RNA. The messenger RNA exits the nucleus through nuclear pores and gets into the cytoplasm, where, with the aid of ribosomes, small and large subunits of ribosome, the process of translation or protein production will happen. And the cells, for their growth and division, they will use a system called cell cycle. We have 5 different stages or phases of the cell cycle. These include G0, G1 or growth 1, S phase where the DNA synthesis happens. G2 is where the cell is preparing to basically divide into two, and M is the mitotic phase where one single cell becomes two cells. Next slide talks about cellular DNA replication, where we have double-stranded DNA, the parental DNA, and then through the action of DNA helicase, DNA unwinds. One single strand basically serves as the leading strand, the other one serves as a lagging strand. Lagging strand produces Okazaki fragments. So there is a link to actually more than one video clip on the process of cellular DNA replication. Again, this is part of the material covered in the prerequisites. If you feel the need, I would strongly encourage you to go ahead and refresh your memory by listening to those and watching those videos, or you can refer back to your old notes to catch up. Basically, the process of cellular DNA replication is where you start with double stranded or positive and negative DNA strand, and you make additional double stranded DNAs. The next process is called cellular transcription. This is the process of double stranded DNA being converted to messenger RNA. This messenger RNA is monocistronic. One messenger RNA codes for one protein. Messenger RNA gets into the cytoplasm where it matures by a 5' 7-methyl guanine cap being added to it, and at the 3' end of it, a poly A tail will be added. These two post transcriptional modifications result in the stability of the messenger RNA. And last part of the central dogma of molecular biology is the process of cellular translation, where the messenger RNA, a messenger RNA that has poly A tail and 7-methyl guanine cap at its 5' tail, will get between the small and large subunits of ribosome, and for each 3 nucleotide, 1 amino acid will be added. And that's called, usually in eukaryotic cells, the first amino acid is AUG or methionine, and the other amino acids will be added. So we will have a growing polypeptide chain or protein. There are some modifications that can happen after production to both messenger RNA and the polypeptide chains or proteins. And these modifications for messenger RNA happen after it's being transcribed. So we call it post transcriptional modification. These include the addition of a poly A tail, so called polyadenylation, as shown in the slide. Polyadenylation, a series of adenine nucleotides will be added, or splicing, where one messenger RNA undergoes a process called alternative splicing, where introns will be removed, and it results in the production of more than one protein by one messenger RNA. We'll talk about alternative splicing later on as well. By the same token, after the production of proteins or translation of the message in the messenger RNA, we will have some changes. These are called post translational modifications. Post translational happen on the polypeptide chains or proteins, whereas post transcription happens on the messenger RNA. Post-translational modifications usually happen in the endoplasmic reticulum or Golgi apparatus, which is a system where the protein is being packaged and getting ready to be sent to its final destination. These modifications include addition of a glucose moiety, glycosylation, phosphorylation. We have other modifications such as acetylation and methylation, ubiquitination. These are some of the post translational modifications that happen to the proteins. The reading material for this, the material that we just covered, came from chapter 2 of the textbook, and some information from chapter 13, where viruses use cellular processes to express their genetic materials. The summary of the lecture: a virus is a biological agent that uses host cell metabolic machinery for its genome replication. Depending on the relationship between the host and the virus, infections can be asymptomatic, acute, persistent, chronic, latent. uh, Chronic and latent are acute, sorry, as well as transforming. And the human host has many sites for viruses to gain entry or penetrate into the host cell and cause infections. The next topic that I want to touch on is the process of virus disease. And the very first aspect of virus disease is the transmission of the virus. Transmission can happen through human to human, it can be through respiratory, saliva, blood transfusion, injections, fecal oral route, or venereals through sexual activity. Or it can be through animals to human, whether and a vertebrate is serving as a reservoir and directly bites a human being, they get infected, or sometimes individuals get virus infection through biting of anthropod vectors, or sometimes there is a relationship between a vertebrate reservoir and an arthropod vector, and then either one of these two, either the vertebrate or the reservoir, can infect individuals. An example of this transmission is smallpox virus, that the vector or route of transmission is through aerosols. Host is human, so human can also serve as a reservoir for the disease and can easily, through human to human infection, pass it on to other individuals. And the disease caused by smallpox is called variola. So this pox virus infection is there is a mouse model of it, which serves as a very good tool to study viral pathogenesis. So what will happen is that they initially inject the virus into the host through the mouse's footpad, and then we follow the infection to see what will happen. As we've mentioned earlier, there would be a lag period or incubation period where the virus needs to successfully gain entry and basically, due to its tropism, go to the tissue that it can cause infection. And this infection is usually countered by the host producing antibodies from its plasma cells to fight off the infection. Then there would perhaps be some symptoms, and the sites of infection or initial infection are skin and the lymph nodes, where we see early signs of the disease, some rashes, etc. Then virus gains entry into the bloodstream. The presence of virus in the bloodstream is called viremia, primary viremia, where the virus from the original site of infection, in this instance skin and lymph node, can infect other organs. So we see some signs and symptoms of the primary viremia. Since virus is in the bloodstream, after infecting the original sites, it can cause secondary viremia from those tissues and organs. It again gets back into the bloodstream, causes secondary viremia, and secondary viremia is where the virus severely infects the skin and causes very severe rashes. So this is a good model for studying disease pathogenesis. Pathogenesis is the process by which an infectious or a disease causing agent causes damage to the host cell. So, depending on the interaction of the virus and the host cells, there would be some outcomes. One outcome of the interaction between virus and host is virus leads to the extinction of the we will have virus extinction, where initially there would be large number of patients getting infected and dying, but those remaining individuals will clear the viral infection and they will acquire immunity. The body develops immunity against the virus. In small populations, this type of infection favors persistent virus infection, meaning that there would be always a little bit of an infection in a small percentage of the population with with those people whose immune response hasn't been mounted against the virus, or immunologically naive persons. Example would be herpes simplex virus, where there would be a transmission of the virus from parent to newborn. However, in large populations, we have a lot of people, because the patient number is higher, we have a lot of individuals who are susceptible to the infection. Therefore, the virus infected individuals are available at all times. For example, a common cold that we see, a common cold occurs in large populations. A lot of individuals get sick, and during the winter time, you always see individuals who have common cold. So it would be a sporadic spread of the virus. This is a redundant slide which only points out to the importance of it, called patterns of virus infection. As we mentioned, four different patterns where a virus exhibits its diseases. It would be either asymptomatic infection, that there would be no disease symptoms; acute infection, the signs and symptoms of the disease are present. The next category would be persistent or long term infection. This can be chronic, where we have infectious virus going on, uh, or latent, where there is no viral replication. After the initial infection, resides in some particular cells. And if the patient has issues with their immune system, or so-called immunocompromised, or hormonal imbalances, or other stimuli, virus becomes reactivated. And lastly is transformation, where the virus modifies or deregulates normal host cell processes. This results in the formation of tumors or cancer. During transformation, there is no infectious virus, so there is no active viral infection going on, whereas we have either parts of or the entire viral DNA present in the host and has been incorporated into the host cell genome. There are viruses whose genome is DNA. We call them DNA virus infections. These DNA viruses can cause acute infections such as HSV, HCMV, VZV, polyoma, HSV, papilloma, etc. Or they can cause chronic infections or cancers called carcinoma. There is a list of viruses which can cause DNA viruses that can cause chronic infection and cancer, such as papilloma, uh, HCMV, VZV, polyoma, HSV, or some viruses are known to cause very severe diseases, maybe in immune naive newborns, those newborns or babies that their immune system has not been fully developed, or in immunocompromised individuals, people who are elderly or they're receiving chemotherapy, etc. By the same token, we have viruses called RNA viruses, that their genome is RNA, and they can cause different types of infections. These include acute infections with specific examples of the viruses that can cause it, chronic infections or carcinoma. There are different viruses, RNA viruses such as polio, hepatitis, rubella, mumps, or they can cause severe diseases such as RSV or respiratory syncytial virus, poliovirus, mumps virus. These are viruses that their genome is RNA and they can easily cause very severe infections and diseases in immune naive newborns or immune compromised adults. An example of an acute infection is caused by varicella zoster virus or VZV. That's the name of the disease, the name of the virus, the causative agent, varicella zoster virus. The pattern is acute infection, and the disease is called chickenpox. So, VZV belongs to herpes virus family. It's one virus that causes two diseases. Varicella virus. It's a common disease of the childhood, and the primary site where the virus causes infections is in mucosa or skin, where the virus causes primary acute mucosal or skin infections. The disease usually resolves within a couple of weeks. However, the virus will remain latent in the nerves. In nerve cells, virus remains latent. So, varicella virus causes skin or mucosal infection. Disease goes away. However, the clinical signs and symptoms of the disease will go away. However, the virus remains latent in nerve cells, which leads to a persistent latent infection called shingles. Shingles is caused by the zoster virus. So we have varicella zoster virus. Varicella causes chickenpox. And then here we have the zoster virus, where the virus remains latent in nerve tissues. We do not have any active infection going on, but we have the presence of viral DNA. Usually, nothing will happen because virus is held in check by the host cell immune defense mechanisms. These can be in the form of humoral immune response or cellular immune response. However, later on in life, the virus becomes reactivated. So it's like it's dormant, it's like hibernating, then it gets up. Once it's awake, it starts replicating, making more of itself, and it descends down the nerve tissue and multiplies in skin cells. And it causes these really painful blisters, and the disease is called shingles. So, the summary of the lecture so far, there are the factors that are implicated in virus infection and disease production are reservoir of the virus, transmission of the virus, and pathogenesis of the virus, or the disease causing capability of the virus. Pox virus was a disease that they've developed a mouse model of it. And the study of this mouse model of pox virus helped tremendously to understand its initial infections or primary viremia, systemic spread, and disease processes. Remember, we said that during this disease, we have two episodes of virus being in the bloodstream, or viremia, primary viremia, and secondary viremia. There are DNA viruses such as HSV, H, HSV herpes simplex virus, HCMV human cytomegalovirus, VZV varicella zoster virus, polyoma, papilloma, hepatitis B virus, or HBV. By the same token, we have RNA viruses like Ross sarcoma virus, measles, hepatitis A virus, hepatitis C virus, rubella, polio, and mumps. The reading for this section of the lecture comes from chapters 3 and 4 of the textbook called Virus Disease in Populations and Individual Animals, and chapter 4, Patterns of Some Viral Diseases of Humans. And lastly, we have uh, discussion questions that I would recommend you guys to go over it for your own practice and make sure you understand the topic a little bit better. Thank you very much for your attention. This is the end of lecture number 2, Virus Diseases.